All right. For our next talk, we have Noé. I'm hoping I'm saying your name right, Perron. He is going to discuss with us the ice plant genome and a little bit different than what's on there. A single nuclei transcriptomic study of the C3 to CAM transition in the ice plant provides insights into key regulators of CAM introductions. He's at the University of Florida. Please take it away.
Thank you. Thank you very much for providing this opportunity and this great platform for both speakers and attendees. Good afternoon, everyone. I'm very excited to talk today about our single nuclei transcriptomic study of the C3 to CAM transition in the ice plants. It is a fascinating topic, and to understand better what all that means, I will start by giving a bit of background information. The increasing occurrence of drought events threatens the sustainability of our agriculture. While a very short-term solution to that issue could be to increase crop irrigation, this practice will only further aggravate the current freshwater crisis. In this presentation, I want to show that we can fix this and without using more water. To find an appropriate solution, we need to look at what is the main driver of economic productivity in the farm. I am talking about photosynthesis. The vast majority of plants on Earth perform C3 photosynthesis. It is a pathway that is characterized by the opening of stomata during the day, which lets CO2 flow inside of the mesophyll cells, where it can be carboxylated by rubisco and then fixated inside of the reductive pentose phosphate cycle, which ultimately leads to the production of photosynthetic sugars. And during the night, the stomata of C3 plants close. The issue with this pathway is that it does not provide plants the capacity to survive drought and conserve water. However, some plants like cacti and pineapple evolved a new mode of photosynthesis called the crassulacean acid metabolism, or CAM, which allows them to conserve water and survive long periods of drought. CAM plants can achieve that by opening their stomata during the night when the evapotranspiration rates are the lowest, so CO2 can flow inside of the mesophyll cells and is carboxylated, this time by phosphoenol pyruvate carboxylase. This reaction results into the accumulation of C4 acids, which are then stored inside of large vacuoles overnight. And during the day, the stomata of CAM plants close, so this helps CAM plants to keep water inside. However, CAM plants are very hardworking. They work both the night and the day shifts. During the day, the C4 acids are transported to the cytoplasm to be decarboxylated, and this reaction provides the substrate CO2 for fixation inside of the reductive pentose phosphate cycle, and the cycle continues. We know that CAM plants achieve 3 to 6 times the water use efficiency of C4 and C3 plants, so a major objective for the next years could be to introduce CAM traits into C3 crops to improve their drought tolerance. Okay, now that we've covered the details of CAM, it is time for me to introduce Mesembryanthemum thymum crystallinum, or the common ice plants. And it is so fascinating because it is a facultative CAM species that can shift from C3 photosynthesis to CAM when it is subjected to salt or drought stress. We know that this transition is initiated after 8 days of salt treatment. However, we don't know much today about the main regulators of this transition. This is why the question that drives my research is the following: can we identify the regulators of CAM induction by single-cell lineage analysis? The best way to answer that question is probably by using single nuclei RNA sequencing tools. Our hypothesis is that we can use the potential of these tools to capture cells at different developmental stages. And by applying this to the C3 to CAM transition in the ice plants, we could be able to end up with a cluster of mesophyll cells in their C3 state and another cluster with the same cells, mesophyll cells, in their CAM states. And by following the trajectory of nuclei cells going from one cluster to the other one, we could be able to identify the key genes that are involved in the CAM induction process. Okay, now that the objectives are clear, let's get into the experiment. We watered 4-week-old ice plants with 0.5 molars of a common nutrient solution, and watered the same number of ice plants with the same solution, but with the addition of 0.5 molars sodium chloride to induce a physiological drought and so to trigger CAM. After 8 days, we harvested the leaves from ice plants in both groups, both after 1 hour into the light phase and 1 hour into the dark phase. So we had a total of 4 samples. We then isolated the nuclei from these 4 samples and loaded them on the 10x Chromium platform for single nuclei RNA sequencing. The data was then processed with Cell Ranger, and we analyzed it with Surat, with Ask Surat, which is a web-based application using Surat. And the output of these analyses are these UMaps. UMaps are great graphs, but they really only become informative when we become able to assign each cluster to a cell type. So how can we do that when we're working on a system that is not very well known, like the ice plants? Well, the first step is to find some known cell type specific markers from the literature, and preferably from closely related species. Then we need to find the orthologs of these genes in the ice plant genomes, if they exist. And this enables us to monitor the expression of these genes in our single nuclei datasets. And finally, we need to make a decision based on the specificity of the marker. In most cases, as you can see in the example on the figure at the center of the slide, the marker is not specific, meaning that it is not specific to only one cluster, in which case we just move on and do not keep the marker. But in the case in which it is specific, like in this example, a cell type can be assigned to a cluster if several other markers that are specific to the same cell type map to the same cluster. So using this strategy, we annotated our UMaps and found some of the major leaf cell types, like the spongy and the palisade mesophyll cells, or the guard cells, for example. And we repeated this exercise for a total of 4 times to annotate the UMaps of each of our 4 samples. So that is a lot of data. There is a lot of information here. So to make it a bit more simple and to answer the question we asked at the beginning of the presentation, we are going to integrate everything into one data set. And this is what we have here. On this UMap, you can see all the cells combined from all the 4 samples. Using the same annotation methods as we described earlier, we found that these clusters correspond to the mesophyll cells. And because the bulk of photosynthesis occurs in these cells, and this is what we care about, we isolated these clusters and rerun the data processing steps to recluster these cells. This is what we can see here. So I know I've been showing a lot of UMaps, but this one is very important. Here we have the mesophyll cells from all 4 samples combined. So it would be very interesting at this point to look at how some of the main genes of the CAM pathway are expressed here. So let's start by taking a look at how phosphoenol pyruvate carboxylase 1 is expressed. It is the main CAM gene, and as you can see in the figure that just appeared at the bottom right of the slide, it seems like the expression of PepC is mostly localized to the clusters at the left end of the UMap. It is a bit more obvious on this dot plot. We can see that the expression of PepC is very high in clusters 0 and 4, which are at the left end of the UMap, but almost negligible in clusters 5 and 6, which are at the right. The same observation can be made for the expression of phosphoenol pyruvate carboxylase kinase 1, which is the enzyme that activates PepC at night. But if we look at the expression of carbonic anhydrase, which is a commonly used mesophyll marker, meaning that it is mostly expressed in the mesophyll cells of almost all plants on Earth, we can see without surprise that it is expressed in a very homogeneous fashion in every mesophyll cells across the 4 samples. So at this point, we made the hypothesis that maybe on the left end of this UMap, we had clusters corresponding to CAM mesophyll cells, and on the right end, clusters corresponding to C3 mesophyll cells. To dive a bit deeper into this question, we ran some trajectory analysis, thinking that if indeed we have some C3 and CAM mesophyll cells, we should be able to see a trajectory, a cell lineage going from the C3 clusters to the CAM ones. So we ran trajectory analysis on this UMAP. You can see these trajectories on the figure that just appeared at the left. And indeed, we found that there is one cell lineage going from cluster 5 to cluster 4, and it takes the following path: cluster 5 to 1, 1 to 10, 10 to 3, 3 to 0, and 0 to 4. So this is very important because it means that by looking at the genes that characterize this trajectory, we may be able to find some of the main regulators of the CAM induction. And this would be a huge step towards introducing CAM traits into C3 crops to make them drought tolerant. So this is where we are at right now. We are about to look at these genes. So I'm going to be leaving you on a cliffhanger for now. But in conclusion, we showed here that the ice-plant single-cell transcriptome provides the keys to achieving a broad understanding of the CAM induction process. An important next step will be to validate the cluster notation via Gus staining. This should potentially confirm the existence of the C3 and CAM cell clusters and will provide a definitive atlas of ice plant leaf cells. And finally, further trajectory analysis may help identify key regulators of CAM induction. I would like to say a big thank you to everyone in the Forest Genomics Lab at the University of Florida for their help and support, and of course to the Plant Cell Atlas Symposium for having me here today. Thank you. I'd be happy to answer any question you may have.
Excellent talk. Very fun and informative. I really liked your presentation. Yes, Kin has a question. The experiment didn't have a time course. Why do you think you saw a trajectory?
The experiment does have a kind of time course. We have, I'm going to go back to the slide with the experimental design.
Yes, please do.
All right. Sorry, here we are. So we, this is, okay, yeah, we have the control group and the salt-treated group. So we have basically ice plants in the control group that perform only C3 photosynthesis at this point. And in the salt-treated group, we have here plants that perform in part CAM photosynthesis. So because of the nature of single nuclei RNA sequencing technologies, we would have some kind of a time course, even if we were just looking at the salt-treated samples. But because we've integrated the control samples with the salt-treated ones, we can really see the progression between the C3, really like the C3 clusters, the C3 mesophyll cells, and the CAM ones. And so potentially, we captured all the cells in the salt-treated samples that go through this transition. And we chose 8 days actually, because it is really the onset of the transition. So cells at this point should be really just starting to perform CAM. So our idea is that we would capture in the salt-treated samples cells in every state of the transition from C3 to the early CAM stages.
Okay, excellent. Daquan Ko has a question for you about, thank you for sharing the great work. I was wondering if you consider the diurnal nature of photosynthesis genes in your sampling and data analysis, since many photosynthesis genes have different activity peaks during the day and night, the cell annotation may be changed.
Yeah, absolutely. And this is actually even more relevant with CAM photosynthesis, because most of the CAM genes will be highly expressed at night, when most of the C3 photosynthesis genes will be expressed during the day. So I've shown here one example of integration we've made, which is the one with all 4 samples together, and which is the most interesting one. But obviously, we are also integrating the samples differently. We're integrating the controls together, the salt-treated samples together, the day samples and the night samples together. And this provides a more accurate comparison. But to really what I wanted to focus on in this presentation was the existence potentially of C3 and CAM clusters. And this we can only see in the integration with all 4 samples together. But yes, absolutely, this is actually a very interesting point and a very interesting thing to look at.
Okay, excellent. Vimmels says, beautiful slides and talk. Is there a specific reason to use single nuclei rather than single cell sequencing? As single nuclei approaches don't include chloroplast genome, which could influence the C3 to CAM transition. What are your thoughts on that? Thanks.
Right. So thank you. We've actually tried first to do single cell sequencing, so using protoplasts, but we found that the bias introduced by using protoplasts is actually more of an issue than anything else. Many salt stress genes are actually induced by the protoplasting, and so it would introduce a bias for the analysis of our data because we deal with salt stress. And we found that single nuclei RNA sequencing is easier. We recover more nuclei than cells when using the protoplast, and we don't have this issue with protoplasting.
Okay, excellent. So in the interest of time, if you wouldn't mind answering Andrew's question in the chat. But thanks again for your talk. It was a wonderful talk. We'll go ahead and move on to the next speaker.